All right, next up's the endomembrane system. Now, the endomembrane system compartmentalizes the cell so that way particular enzymatic reactions are going to be restricted to specific regions and help increase the overall efficiency of the cell. When we restrict those enzymatic reactions to specific compartments, we can prevent incompatible reactions from happening at the same time or in the same location. We may have, coming out of our ribosomes, we may have new proteins being synthesized. We certainly don't want those right next to enzymatic proteins that are going to start breaking those proteins back down again. So we just want to make sure that we kind of help keep everything organized as efficiently as possible. The endomembrane system is going to consist of the nuclear envelope, the membranes of the endoplasmic reticulum, the Golgi apparatus, and then also vesicles. The endoplasmic reticulum, it's going to be a series of membrane channels and saccules. Saccules are just like flattened vesicles that are continuous with the nuclear envelope. So I know how we looked at the nucleolus and the nuclear envelope in our previous slides. I'm going back here, and it looks like it's all one sort of self-contained region. It's not. This double membrane actually branches out and forms the endoplasmic reticulum as it branches off. So it's a system of membrane channels that is continuous with the outer membrane of the nuclear envelope. Here's what it looks like up close. So we do still have our double membrane, that is our nuclear envelope, but branching out from there, here's a nuclear pore, but branching out from there, we have our endoplasmic reticulum. Now, endoplasmic reticulum can actually be broken down into two categories. The first one is rough ER. This one is named for its appearance. Rough ER is going to be studded with ribosomes. Oh, rough ER and smooth ER. Rough ER, here we go, rough ER. It's named for its appearance. It's going to be studded with ribosomes on the cytoplasmic side, on the side that reaches the cytoplasm and not on the inside. So the inside is nice and smooth, and then there's going to be ribosomes studded throughout the outside. This is going to be a site primarily for protein production, which makes a lot of sense. Our message for how to synthesize a protein, our mRNA, is coming from our nucleus. And guess what? We have one continuous membrane. All we have to do is go out of the nuclear pore right into a ribosome right here, and we're going to start working on our protein production. So rough ER's primary job is going to be the synthesis of proteins. However, not only do we synthesize proteins, we can also modify and process those proteins. Now, if you remember, when we talked about protein synthesis, and like I said, we haven't really covered it yet, but we will later on. The very first level of structure of protein complexity is going to be that polypeptide chain. Now, once we have a polypeptide chain of amino acids, do we have a completed and full protein? No, we do not. All we have is a chain of amino acids. It is not until we modify and process that protein, not until we fold it up into its proper three-dimensional shape, do we actually have a completed protein. That's going to happen inside the rough ER. Not only are we going to make proteins, process proteins, we are also then going to transport those proteins. We're going to stick them inside transport vesicles to be shipped off to the rest of the cell or wherever it is that they need to go. So this is kind of like, well, I don't know if any of you have ever worked in a factory before, but when I was in college, my summer job was working at a junk mail factory. At least that's what I used to call it. It was technically a paper mill, but we didn't just make paper. We printed it and cut it and made flyers, and we essentially made all of the junk mail and flyers that you receive every day. Cut it and processed it. So that mail or that paper was made, and then it was processed through the mill. It was processed down the printing press, and then we cut it, fold it, and then we prepare it to be shipped to the post office. So we would stack it up on pallets, we would send it off to the side, mark an invoice, and then it would be sent off to the U.S. Post Office. From the U.S. Post Office, it can then be distributed countrywide. So that's going to be sort of the role of the rough ER. It's going to produce those proteins, it's going to process those proteins, and then it's going to get them ready for transport. Now, there are two types of endoplasmic reticulum. We've only talked about the rough ER and its role in protein synthesis and processing. The smooth ER is going to be very similar, but it doesn't have ribosomes. Because it does not have ribosomes, it is not going to play a role in protein synthesis. Instead, it's going to be the site of lipid synthesis. This is where we're going to be making our fats and oils. This is also going to be the site of various synthetic processes, detoxification, and storage. Liver cells are going to play a huge role in detoxification of the body, so the liver cells tend to contain a lot of smooth endoplasmic reticulum. This is also going to form transport vesicles. Just like the rough ER produces, synthesizes, processes, and gets and packages proteins for shipment, smooth ER is going to do the exact same thing, but for lipids. Now, once we have formed our transport vesicles, we need to then ship those vesicles. Now, the ER isn't really responsible for directing where those packages go. We're going to form transport vesicles and we're going to send them off to the Golgi Apparatus. The Golgi Apparatus is the U.S. Post Office. It is going to be responsible for tagging, identifying, and deciding where each one of those are going to be delivered. So this is going to be a series of flattened and slightly curved saccules. You can kind of think of it as a stack of hollowed pancakes. It's going to lie between the ER and the plasma membrane, which makes a lot of sense because the ER, the endoplasmic reticulum, is where our proteins and our lipids were just freshly made. Now they need to be shipped out. Some of those proteins and lipids are going to be processed and used in the cell. Some of them need to be excreted. For example, we've already sort of mentioned our pancreatic cells who are responsible for producing insulin. Those cells that are producing insulin are not producing insulin for the sake of the cell. They're producing insulin so it can be sent to the bloodstream and can be responsible for picking up or for absorbing glucose or releasing glucose into the bloodstream. So we're going to be producing hormones that are going to help regulate blood glucose levels. So the whole point of producing insulin is not to just keep it stored up in our cells, but to release it. The Golgi apparatus is going to be responsible for doing that. The proteins are going to be made by the rough ER. They're going to be sent to the Golgi apparatus, and the Golgi apparatus is going to figure out where they need to go. Do they need to go somewhere inside of the cell, or do they need to be brought to the cell surface? So as I said before, it's a series of flattened curved saccules, kind of like a stack of hollow pancakes. They're going to be modifying proteins and lipids with signal sequences. Just like when you take a package to the post office, they're going to slap a shipping label on it. They're going to figure out where it needs to go, scan it into their system, and send it on its way. Same thing's going to happen with the Golgi apparatus. We're going to modify the proteins and lipids with signal sequences. Those proteins and lipids, they're fully formed. That protein is the protein that it needs to be. All it needs is a shipping label. And that's what we're going to get from the Golgi apparatus. So it's going to receive the vesicles from the ER. It's going to modify those lipids and proteins, prepare them for shipment, and package those proteins and lipids that leave the Golgi into vesicles that are going to eventually leave the Golgi. So those transport vesicles get picked up here, they get processed and passed over here, then passed to here, then passed to here, then passed to here, then passed to here. And once they finally reach the end, they're going to be put into transport vesicles, fully modified and ready to go. Here's an overview of the endomembrane system. This is a fantastic slide to quiz yourself. See if you can recognize all of the shapes and organelles. See if you can identify everything that's going on in this image. Where's the nucleus? Where's the rough ER? Where's the smooth ER? Where is the site of protein synthesis? Where are the lipids coming from? How are they being transported into and out of the cell and through the cell? So definitely spend some time with this diagram. If you can talk yourself through everything that's going on in this image, you will have a fantastic understanding of the endomembrane system.